thank you, Lionel, for inviting me for this opportunity to show the pair correlation approach at this workshop. Is now tradition for us to be speaker and invited at this fantastic workshop. And yeah, I don't think I can hear you, but please write in the chat if you have any questions. Stop anytime. Today I'm going to show you the pair correlation approach and why it's so powerful, and it's important to understand the biophysics of complex environment. Okay, awesome. Thank you, Andres. So this is the outline of the presentation. So first we are going to show you 2 minutes about history and review of FCS, why it's relevant to this talk, and then the pair correlation concept, that is a concept that's been developed by Enrico Gratton and Michel Digman, and is very powerful. I'm going to show you a couple of examples for the application of this technique and why it's so powerful. And at the end, yeah, these approaches, these applications have been published. So yeah. So FCS, I think you people, you already could see.
Yes.
Any issue? I cannot hear you, Lionel.
I can see you slide.
Okay. Okay, so yeah, at the beginning, the fluctuation where the fluorescence fluctuation were used to understand the chemical reactions, and these fluctuations were, the interpretation of the fluctuation were used to understand and characterize fluctuation of chemicals during reactions. And then after, from the 70s to middle 70s, the particle interpretation of FCS has been proposed, which I think is the one that we in some way we like to think about. And then the fluctuation, the description of the fluorescence fluctuation to describe. Okay, the fluctuation from the same molecules, and then spatial correlation for FCS was developed a few years ago with Michel and Enrico in 2005. So the spatial correlation, PCF, is one of classical example of spatial correlation, or better, spatio-temporal correlation. Okay, so why spatial FCS and spatial correlation? The idea of using spatial correlation to understand diffusion reactions and to have an idea of the biophysics of the system has been already described before this morning, and you could see from Lorenzo Scipioni talk RICS. So FCS in some way is another way to characterize the spatio-temporal correlation. What is important, this tool is very important for the characterization of barriers, and I'll show you why. And in some way can overcome the limitation of single particle tracking, because single particle tracking works only when you have very few particles and you can monitor the dynamics of very few particles. For PCF, you can have a multitude of particles, so you statistically will understand the dynamics of the particles. And this is why you know from this morning the concept of point spread function. So basically the point spread function is this physical volume of light where the particle diffuse in and out, okay? And when the particle goes in, the fluorescence is turned on and you have the fluctuation thanks to that. And with single point FCS, this is the classical FCS experiment. This is the correlation function. So you basically you correlate the shape of the fluctuation over time, over the delay time. And what kind of information can you get from this technique? The diffusion coefficient and the concentration of particle. But what happens if instead of having one FCS, point spread function, we have two point spread function. So we can apply the same formula and we can correlate the fluctuation between the first point spread function and the second point spread function. So we can cross correlate between these two sampling regions, I would say. And this approach has been shown and published by Petra Schweil in 2006. So we can use, so here in this setup, we have two physical point spread functions. We can use the same concept, or we can change the concept. We can change the approach simply moving the scanner. So if we move the scanner, we move the point spread function, and we can sample over time the correlation between these different point spread functions. Okay, why is important? Because we can use this approach to visualize an image and characterize obstacles and barriers. So every circle here is a point spread function. Imagine that we are sampling the space, moving this point spread function along these lines, along this axis. And if we have the particle, for instance, the particle in position 1 that is interacting with this barrier and then going back, we might not see any correlation across the barrier. While if it is an obstacle, maybe after a certain period of time, we can see the particle, we can have positive correlation of particles if they can surround the obstacles. So with this approach, with the pair correlation concept, we can distinguish whether it's a barrier and whether it's an obstacle. And sometimes in biological systems, the difference is that maybe a short period of time, the structure is a barrier. Over a long period of time, maybe it's an obstacle. Okay, so we can characterize the traffic and the dynamics in complex system. Okay, so the idea is to measure the correlation across space, moving the point spread function, and we can cross correlate. So now the question, are we sure that we are seeing the same molecule, that we are correlating the same molecule? Because it's not trivial to trust this point. So during this presentation, I'll show you why we are actually sampling the same molecule that is moving across the space. So the pair correlation concept. If we have a particle at time 0, position 0, we know that the probability of finding the particle at a given distance depends on the intensity of the particle at time 0 and position 0. And this probability decay over time across this function with this trend, basically. So we have the intensity time 0, position 0, and we can monitor on the same position over time, and the probability of having the same particle in this point spread function is decreasing over time. So after a while, the particle is not anymore inside at the same spot because the particle is diffusing. If we change spot and we check, we can see that the particle is appearing after a certain amount of time, and the amplitude of this, it depends on the intensity at time 0. So what's the interpretation of this plot? That this curve here is the classical autocorrelation function curve, where it's telling the average amount of time that each particle is spending in the point spread function. If we check this, we have a distance, okay, and this is giving the time, the statistical time of having the particle from time 0, from position 0 to this point. Okay, so it's basically telling us how much time we need to wait to have the particle in this position. Okay, so we can use this approach to image and characterize barriers and obstacles, as you can see. Okay, and I'll show you why, and I'll show you how we can use this to follow the same molecule, the same particle statistically. Okay, so the idea here is we collect the fluctuation on a scanning line over and over and over, and then we can get the fluctuation position 0, position 1, over time, and we can cross-correlate the fluctuation with the fluctuation recorded at a given distance. So the autocorrelation function becomes in this way. So we add the term is basically these terms, it gives us information about the space difference between position 1 and position 2, okay? And the idea here that we are correlating the fluctuation over time at position 0 with the fluctuation over time at position delta r, okay? So if we have the particle that is diffusing inside this point spread function and is flowing in the second point spread function after a certain period of time, we have positive correlation, okay? And the mathematics explain why we have positive correlation only when we have the same particle, okay? Because we have positive correlation only when the fluctuation are matched in shape, in basically width and height, okay? So we can simulate that. We can use not only line scanning, we can also use orbital scanning, okay? So the idea here that we have over time, okay, and we are checking the correlation of particles. This is the points along the orbits, okay? This is depending on the concentration. But basically, point correlation 1, it means that we are shifting the correlation by 1 pixel. And usually the pixel are 50, 60 nanometers, just to give you a reference, okay? So if we have the two point spread function overlapping, we are still sampling the same autocorrelation, okay? So we have pair correlation when the two point spread function are not overlapping anymore, okay? So PCF1 is still autocorrelation. PCF2 is shifting, so is less autocorrelation, it starts to be more pair correlation. When we have two completely different pair correlation function, in terms of we don't have point spread function, they are not overlap, these focal volumes, we have pair correlation function. Okay, so the decay and the trend depends on the diffusion coefficient. Okay, this is important. So the idea here that is we have the particle at position 0 that is diffusing, and we are scanning, and we can find probably after a certain period of time the particle that can give us positive correlation. So the main question is why we have 0 or negative correlation in the pair correlation function. And this is exactly the reason why that explain why we are seeing the same molecule, because it means that at times, like a short period of time, we don't have correlation. So this means that we have a positive correlation only at certain amount of period of time. So this means enough time for the same particle to move from point spread function 0 to point spread function after a certain period of time at given space, okay? So this is the reason why we that tell us that we are sampling the same particle. And the shape of this, it depends on the diffusion type, and depends also on if we have 2D or 3D types of diffusion. And the amplitude depends on the type of diffusion. So the amplitude of the pair correlation depends if it's 2D or 3D function, 3D diffusion, sorry. So we can, so far we have seen simulations, and here we have a real example. So these are beads in solution that are diffusing, okay? If we sample the fluctuation and we do the correlation function, this is the ACF, okay, basically this trend, and we can fit this trend with ACF correlation function to measure the diffusion coefficient and the number of particles. If we check the correlation at a given distance, and this given distance is much larger than the point spread function, we have PCF at 6 pixels, And we can see that this depends on the diffusion coefficient of the particles. So you can actually, if you have SIM-FCS, you can actually simulate this system, and you can actually do the same experiment in silica, okay? So the idea here that we are scanning on a line with a laser scanning confocal microscope, but we can actually use also camera system. We can scan it several times for several lines and then apply this algorithm, okay? So what's the difference between RICS and PCF? So usually RICS requires a much slower type of scanning, while PCF is basically the fastest you go, the better, okay? There is some trade-off for signal to noise ratio, and this depends on your system, but this is more or less the golden rule that makes the difference between RICS and PCF in terms of instrument settings. So one of the I think the most famous example, and I think that can show how powerful and how can we make an interpretation of the data that we obtain from this technique, is the diffusion of GFP inside the nucleus in the chromatin, okay? So here we have the particle, okay, and we have PCF, and the particle, so chromatin is characterized by regions of high density and region of low density. Okay, so if we monitor the particle inside the low density region, okay, we can see that this particle cannot go through high density region. And here we have no communication. No communication means there's no correlation, and there's no way that this particle can overcome this barrier at this distance and this time, okay? But if we increase the distance or we increase the amount of time, we can actually see the correlation of particle going in and out, or the particle going across the from low density region to high density region. So we can map the chromatin accessibility that is crucial for epigenetic, and we can information about the DNA structure and again all the transcription machinery that is occurring inside the cell. Okay, this example has been published on PNAS, I think 2008 or something. I think the first author, Elizabeth Hind. I think if you want, I strongly recommend to read this paper because it's very beautiful and elegant. Okay, and it gives us the possibility to understand how we make an interpretation of this data set. We have these carpets, and we can make arch on this carpet. And when we don't have connection, like in this case, means we have a barrier. If we have a connection, it means that after a given period of time, the particle can go from this pixel to this pixel, okay? So what's the classical example and for the description and to demonstrate how powerful is this technique is the description of the nuclear pore complex. Nuclear pore complex is a structure is very complex because it's basically orchestrating the traffic between the cytoplasm and the nucleus. So it's responsible of the shuttle of molecules across the nuclear membrane. So it's important for everything that involves genetics and protein expression. And there are many diseases that that have like problems and issues on this structure. Okay. Why is this interesting? Because nuclear pore complex is a filter for sites. It's a size filter because molecules that are larger than 60, 70 kilodalton cannot go through. Okay, so there could be a passive diffusion for small molecules, and then also an active transport because there are shuttles that can move across this pore complex, okay, and can move big molecules across this structure, okay. And usually the active transport is carried on by nuclear locating signals and sequences. Those are short peptides that binds the that binds the molecule, and they can orchestrate the shuttling between. And is most of the time is ATP dependent transport. So we have the passive diffusion and active import that we can discriminate and characterize with PCF. So what's the idea here? We have GFP NLS that can interact with importing alpha and importing beta. Importing alpha and beta can move this huge molecule across the nuclear pore complex, but this molecule is small enough to have also and exhibit passive diffusion. So we can use this system to check and understand the differences between, and you can use PCF to characterize the diffusion across this nuclear pore complex, and we can separate the transport from passive diffusion. So how this system has been characterized before has been characterized using FRAP, fluorescence recovery after photobleaching. Is very invasive. Basically, you bleach all the nucleus and then you observe the recovery of fluorescence. So basically all the molecules that are diffusing from the cytoplasm to the nucleus, and you can have information about how long it takes for these molecules to move from the cytoplasm to the nucleus. And other experiments have been done using single molecules. So basically you have the molecule like that of interest that you want to study, the shuttling across the nuclear membrane, that usually is bound to a very big bead, and you can monitor the fluorescence of the bead across the membrane, but is not that elegant because the bead usually is larger than the nuclear pore complex. It gives information at single molecule, but we can have the same information statistically using the PCF without this limitation. Okay, so no purification, no labeling. We make express the proteins from the cell, and we can start to characterize the dynamics of this system. So what's the idea? The idea here, we have the GFP NLS, and we can characterize the diffusion within the nucleus, within the cytoplasm, and the diffusion and the dynamics across this membrane. You can do this with the camera, but usually to visualize the nucleus, it's way better to use a laser scanning confocal microscope. So we set the line across the nuclear envelope, and we monitor and we acquire several thousand of lines, and we can basically, we have this information. We have the intensity over time. Okay, so we can start to correlate particles in the nucleus, in the cytoplasm, and with a autocorrelation function with ACF. And we can also correlate the particles that moves from the nucleus to the cytoplasm following active and passive transport. Okay, for instance, we can start with ACF. ACF, it means that we correlate the particle in the nucleus in the cytoplasm. So what we see, we can use the ACF to measure the diffusion coefficient in these two compartments, and we can see that in the nucleus is slightly faster than the cytoplasm. This is because in the cytoplasm there are organelles that can slow down the diffusion of this system. And what we see that if we move close to the barrier, we see a dip in the correlation. So it means that something is happening at the nuclear membrane. So the particles stop to correlate, basically. Okay, so we can use the PCF to understand the diffusion across the barrier. And you can see here that the dynamics here is complex when we are in the proximity of the nuclear membrane. So we can characterize the intra-compartment diffusion, and we can set, for example, the pair correlation between pixel 1, pixel 2, pixel 3, pixel 5, sorry, and pixel 13, okay, and we are in the nucleus. We can do at the same thing in the cytoplasm, and we can have information about the correlation. If we cross-correlate pixel 1 with pixel 16, 17, for instance, we can have information about the time that occurs for a particle to go from the nucleus to the cytoplasm. And in this case, we have information about the transport of this system, okay? And this is the time, the correlation time that we observe. So we can use this to characterize the diffusion across these two compartments, and of course is a barrier a short period of time, and this barrier the particles can diffuse across this barrier after a very long period of time, and this is the correlation time, okay? So it's between 100 and 500 millisecond range, okay? So we can characterize all these things. We can also check the presence of the importing system and check the correlation in the presence of import carriers. And we can also deplete ATP to restore the only possible mechanism as passive diffusion and check the diffusion using PCF. One thing that we can demonstrate that this system is actually monitor the right particles is for example monitoring the diffusion across cytoplasm of two different cells, and we don't see any type of correlation. So two cells do not exchange this kind of fluorophore and this kind of molecules, and this is telling us no matter the time we have an impenetrable barrier if we check two different cells, okay? So this is the overview of the system. So we can PCF can measure molecular flow at 100 nanometer scale. Experiment with beads can be reproduced, and simulation and experiment with beads are perfectly in line, and also simulation of like GFP, or the nuclear pore complex system that I just showed you. We can demonstrate how to measure diffusion, passive diffusions versus transport, and we can have a complex information about heterogeneous system, like for example the chromatin. So yeah, I'm happy to answer your questions.